In this video, we will be learning about the electron transport chain. So let's dig upon it. So what exactly electron chain is? So electron chain is meant to produce ATP, that is the adenosine triphosphate basically, which is the energy currency of our cell. So basically the need of this electron transport chain inside the mitochondria is to produce ATP. So from our knowledge we know that in glycolysis NADH is produced even in the formation of acetyl-CoA from pyruvate, that is the end product of glycolysis. And this acetyl-CoA, when comes inside the matrix, it is used to produce the glucose by the Krebs cycle, and in this process NADH is produced, also FADH2. So this NADH and FADH2 during the Krebs cycle are produced and are used to form the ATP. So how does it happen? So let's start this process in a brief, and later we will discuss about the complexes separately. So here, as you can see, this is the inner membrane formed by the bilipid layer, and we have these proteinaceous complexes embedded within the inner membrane of the mitochondria. So beyond that we have the intermembrane space, and inside the inner membrane we have the matrix of the mitochondria. So what happens that in Krebs cycle NADH is produced, and also from glycolysis. So this NADH is oxidized to its NAD plus form, and during this oxidation there is occurring dehydrogenation. As you can see, there is a loss of hydrogen and also electrons. So these two electrons and two hydrogens. So what happens to these electrons and hydrogens? So this electron is transported to a mobile electron carrier. So in this electron transport chain you will basically face two mobile electron carriers. Here this one is known as ubiquinone and this one is known as cytochrome C. We will discuss that later. So what happens that these two electrons is transported to ubiquinone. So the ubiquinone takes up the electron from the complex 1 and transfers this electron to the complex 3. In simultaneous, there is another reaction in the Krebs cycle where there is conversion of succinate to fumarate. So during this conversion, there is liberation of two hydrogens and two electrons, and this electron is again transferred to another molecule of ubiquinone. So ultimately what happens, both these complex 1 and complex 2 gives their electron to the ubiquinone, which is a mobile electron carrier, and this ubiquinone takes up this electron and gives it to the complex 3. So what happened, the electrons are transferred to the complex 3 by the ubiquinone from both the complexes 1 and 2. After accepting these electrons, this complex 3 transfers this electron to the cytochrome C, which is another mobile electron carrier. So the cytochrome C is water soluble, so it jumps up to the complex 4 outside the inner membrane and gives up this electron to the complex 4. So basically what happened, these electrons from the complex 3 is transferred to the complex 4 via the mobile electron carrier, that is the cytochrome C. Now these electrons, after reaching the complex 4, it is used to form one molecule of H2O. So these electrons transferred by the cytochrome C is used to form one molecule of H2O. As you have seen, these electrons are transferred in this fashion of a chain like manner. That is why it is known as electron transport chain. And during this electron transport, due to redox potential, hydrogens are pumped to the intermembrane space, increasing the H plus concentration. That is why it is called the p side, having more positive charge, and it is known as n side because of less positive charge, also termed as negative charge with respect to this intermembrane space. So during the transfer of the electrons, hydrogen ions are pumped to the intermembrane space. This hydrogen increases the H plus concentration in the intermembrane space, and these hydrogens are used to form ATP. So what happens that this ATP synthase, which is the complex 5, takes up 4 hydrogen atom. So it takes 4 hydrogen ions and uses these hydrogen ions to generate one molecule of ATP. So the complex 5 needs 4 hydrogen ions to produce one molecule of ATP. Like that, due to the difference in the concentration of hydrogens, the hydrogen comes to the matrix via this complex 5, and during this transfer of 4 hydrogens, each molecule of ATP is produced from the ADP and Pi. So let's go to the complexes individually. So let's understand the complexes that are involved in this electron transport chain. So in first we have the complex 1 known as NADH dehydrogenase complex, as it is causing the dehydrogenation of the NADH. So I am describing it right now how it is causing the dehydrogenation. So first of all, the complex 1 is a FMN, FeS, iron sulfur, and flavin mononucleotide containing L-shaped proteinaceous complex. So how this transfer of electron works inside the complex 1. So what happens that the NADH get oxidized to NAD plus. So these electrons released are transferred to the flavin mononucleotide, and then the flavin mononucleotides gives this electron to the iron sulfur complex, and from where this iron sulfur complex transfers this electron to the ubiquinone. So the ubiquinone is reduced to ubiquinol by transferring electrons and hydrogens. So we have talked about the complex 1. So how that complex 1 gives its electron ultimately to the complex 3 via the mobile electron carrier, that is ubiquinone. The same happens here for the complex 2 also. So as you can see, the complex 2, given by its name, succinate dehydrogenase complex. So in Krebs cycle, the conversion of succinate to fumarate occurs by this complex. During this conversion, two hydrogen ions released per oxidation of a succinate molecule is accepted by the FAD+, known as flavin adenine dinucleotide. So what happens in Krebs cycle that simply there is a conversion of succinate to fumarate, and during this conversion of succinate to fumarate there is liberation of hydrogen. That is why it is named as dehydrogenase complex, where the dehydrogenation process is occurring. So this hydrogens in succinate is removed and there is addition of a pi bond instead. So these hydrogens are removed by the action of the enzyme known as succinate dehydrogenase. So this enzyme helps in the removal of these hydrogens, and these removed hydrogens are accepted by the FAD plus molecule, converting it to FADH2. So now what happens inside the complex that this FADH2 is converted to FAD plus. So during this conversion it gives up its two electrons to the iron sulfur complex, and this iron sulfur complex gives its electron to the mobile carrier of electron, that is ubiquinone, and along with the addition of hydrogen atom. So this addition of two hydrogen atoms along with two electrons convert this ubiquinone to ubiquinol, which is its reduced form. So it is converted to ubiquinol, that is the reduced form of ubiquinone. Now this ubiquinol produced in both complex 1 and complex 2 are transferred to the complex 3. So let's see what happens next. So as far we have seen that ubiquinone is converted to ubiquinol by accepting electrons and hydrogen ions. So two electrons and two hydrogen ions are accepted by the ubiquinone, which converted it to its reduced form, that is the ubiquinol. Now this ubiquinol comes to the complex 3 where the new story begins. So the complex 3 known as cytochrome oxidase. So the reduced form of ubiquinone. So the reduced form of ubiquinone, that is the ubiquinol. So this is the reduced form of ubiquinone. It releases its two electron to the complex 3. So now what happens, this ubiquinol goes back to its previous state, that is ubiquinone, by releasing its two hydrogen and two electrons to the complex 3. So now it oxidized back to its previous state, that is the ubiquinone, by releasing the two hydrogen ions and two electron to the complex 3. Now what happens, let's see. So this two hydrogen goes to the intermembrane space by pumping mechanism and accumulates there. So these two hydrogen ions liberated goes to the intermembrane space by pumping mechanism. And what happens to the two electrons? So now these two electrons is transferred by the complex 3 to another mobile electron carrier, that is the cytochrome C. So this cytochrome C is another mobile electron carrier like the ubiquinone. The difference is that it carries one electron at a time. So what happens, let's keep it aside. So what happens that the two electrons are accepted by the cytochrome C, which is another mobile electron carrier. So they carry these electrons from the complex 3 and travels to the complex 4 and removes these electrons to the complex 4. Basically what happens that it transfers the two electrons from complex 3 to complex 4. So now let's get on to the complex 4. So the complex 4 known as cytochrome oxidase. So cytochrome C oxidase is the name of the complex 4 where the oxidation of cytochrome C happens. So this cytochrome C, after accepting electrons in the complex 3, get reduced, and now again the cytochrome C releases its two electron and transfers it to the complex 4. So the cytochrome C now again gives up its two electron to the complex 4, and using these two electrons H2O is produced, and during this process 4 hydrogen ions are pumped to the intermembrane space. So what we have seen that the ubiquinone is converted to ubiquinol after accepting electrons and hydrogens from complex 1 and complex 2. So this ubiquinol is transferred to the complex 3, that is known as cytochrome reductase. In the complex 3, the ubiquinol releases its hydrogens, that is the two hydrogens and two electrons. Now this two hydrogen is pumped to the intermembrane space and the two electrons are given to the another mobile electron carrier, the cytochrome C. The cytochrome C now transfers this electron to complex 4, and the complex 4 uses these two electrons to produce one molecule of H2O, and during this process 4 hydrogen ions are pumped to the intermembrane space via the complex 4. So complex 5. So it is our last complex known as ATP synthase because synthesis of ATP happens here. So what we have seen so far that in case of NADH oxidation in complex 1 there occurred transfer of 4 hydrogen ions to the intermembrane space. So in complex 1, 4 hydrogen ions were transferred to the intermembrane space. In complex 2, there were transfer of 2 hydrogen ions to the intermembrane space. And in complex 4, there were 4 hydrogen transfer in the intermembrane space. So this pumping of hydrogen to the intermembrane space increases the hydrogen ion concentration. So this increase in hydrogen ion concentration causes what? So many hydrogen ions are accumulated in the intermembrane space, that is this area. So there is lot of hydrogen ions as you can see, and the complex 5, which is this whole complex, this complex 5 uses only 4 hydrogen ions. So it uses only 4 hydrogen ions to generate a single molecule, only one molecule of ATP from one ADP and one inorganic phosphate. So this complex 5 uses 4 hydrogen ions to produce one ATP molecule. So as you can see, this is the intermembrane space. It is using 4 hydrogen ions to form one molecule of ATP. In for knowledge, this is also called F0 F1 particle. So this part embedded in the inner membrane is called the F0, and this outer part is called F1 particles. This is called the F0 F1 particle, and these are the respective subunits of this whole ATP synthase, that is the complex 5. So the main ultimate goal here is to produce ATP using the hydrogen ions that were accumulated by the action of the pumps used in the previous complexes. I hope you like this video, so please give a thumbs up and do subscribe my channel. In the next video we'll be doing the calculations of ATP involved in the aerobic respiration. Till then, stay healthy. Bye.